Hey guys, Miss Pastek here. So today I'm just creating this video so that you guys have something to refer to if you want to learn a little bit more about Punnett squares. And so we are jumping straight into this unit of genetics. And so understanding Punnett squares is really important, especially when it comes to Mendelian genetics. And so this video, first off, I'm going to show you guys a quick little PowerPoint that I've created that goes step by step through some vocab and then through the process of creating a Punnett square and figuring it out, and as well as understanding probabilities. After that, I'm going to go through the lab that you guys are going to be doing this week and just highlighting some of the things that you guys should really look at in order to understand the lab in its entirety. So hopefully you guys are doing good. Hopefully you're enjoying the nice weather. I know I'm going to go outside after this video. And yeah, so let's just get started. So I'm going to present here. Awesome. Okay, so I've got Punnett squares ready to go. So this just serves as a quick little review. We're going to go over some vocab, then we're going to go over a scenario that I think is pretty relatable to Punnett squares, and then hopefully you guys will feel a little bit more comfortable doing these. So this genetics unit that we're going into is all about genes. And so a gene is a unit of DNA that is transferred from a parent to an offspring. And so we get a lot of our genes, all of our genes from our parents. And so genes are critical for understanding genetics and just understanding that it's a unit, it's a piece of DNA that you get from your mom and dad. That is really important to understand. An allele is one of the possible forms of that gene. And so a gene can take multiple forms. We're going to talk about dominant and recessive alleles. And so alleles are just that part of the gene that takes different forms. Okay, so it's a different form of one's particular gene. Okay, so now we're going to talk about dominant and recessive. So we did talk about alleles. Alleles have two different types of things that they can be. They can either be dominant alleles or recessive alleles. And so a dominant allele, these are going to have more of a say in what the outcome of the cross will be. And I'm sure when you guys went over this in middle school, you remember them being represented by uppercase letters. And so a big T might represent a dominant allele, whereas a little t might represent a recessive allele. And so a recessive allele, these are going to have little to no say in what the outcome of the cross is going to be. And so just like I said earlier, these are represented by lowercase letters. And so you can really choose whatever letter you want in the alphabet to represent a certain trait that you're talking about, a certain gene or allele that you're talking about. But in the case of dominant and recessive, you need to remember that dominant is always associated with uppercase letters, while recessive alleles are always associated with lowercase letters. And so the combination of the lowercase, uppercase letters, the different combinations that you can get will tell us whether or not the individual is heterozygous or homozygous for that particular thing. So heterozygous means that they're two different alleles. And so you're going to have one dominant and one recessive allele that are heterozygous. And so an example of this would be a big T little t, or a big G little g, or a big Y little y. That combination of uppercase to lowercase letters just tells you that there is a dominant with a recessive allele that are together, making that heterozygous. Homozygous is going to be two of the same alleles. And so this can either be two dominant alleles together or two recessive alleles together. So in the case of the letter T, you could have big T, which would be homozygous dominant, or you could have little t, which would be homozygous recessive. So just understanding heterozygous and homozygous can really help out, especially as we get towards the end of Punnett squares and you're making conclusions about the probabilities of certain outcomes. And so now jumping into Punnett squares, what is it and how do we use them? Like, why are they important? These are tools that scientists use in order to predict the genotypes of a particular cross or breeding experiment. So we're going to talk about genotypes and phenotypes in just a second, but understanding that these Punnett squares can help us predict about what the outcome could be between two different organisms. And so you might have a male and female that are mating, and the cross of those will a Punnett square could tell you what the probability of their offspring having a certain genotype might be. And so Punnett squares tell us about the probability of an offspring having a particular genotype. And so genotypes are the set of genes that an organism carries. And so the genotypes are often associated with that big T, little t, or big T, little t, all of those letters that we see in the Punnett square. The letters themselves can tell us about what the observed characteristics might be. So if a dominant allele represents a certain eye color, let's say brown, so let's say big T represents brown eyes, and then little t represents blue eyes. So if you have alleles that are dominant, you're probably going to have an offspring that has a brown eye color. If you have recessive alleles, you're going to see that the offspring will have blue eyes in that case. And so we're going to jump into an example of this and how genotypes really determine the phenotypes of an organism. And just understanding that is really critical and can really help scientists understand what the possibilities could be for that particular organism. And so an example of this is albinism. And so I'm sure you guys have seen that certain organisms have albino characteristics. I know that there are tigers that are albino, but I thought that this little kangaroo guy is pretty cute and worth showing. So albinism is the lack of pigmentation in the eyes, the skin, and the hair. And so this is inherited. It is inherited, resulting from a combination of recessive alleles passed from both parents. And so the genotype that codes for this albinism is little f, little f. So I just chose F as the letter that I want to use. And we know that it's a recessive trait. And so if an organism, if this kangaroo is recessive for FF, it is going to be albino. And so let's create a Punnett square using what we know about albinism. And then we're going to cross some parents and see what the chances are of getting an albino kangaroo. So in the case of albinism, we know that it appears only when the offspring has two recessive alleles for that particular gene. So we've got three options here. We've got big F, which is dominant. And so dominant meaning normal. Those kangaroos look normal compared to an albino kangaroo. They've got the brownish gray fur coat and then look normal in skin pigmentation, etc. So big F is dominant. Big F, little f is also dominant because it has one of those dominant alleles in it. It has the big F. And so the big F, if we remember, the dominant allele is going to override the recessive allele if those two are together. So we've got a homozygous dominant, a heterozygous dominant. So these are both dominant, coding for normal phenotypes. In the case of little f, it is going to be recessive. We know that because it is going to be albino. So if we have a little f, it's recessive, and that particular offspring will be completely white or albino. So what is a scenario that we could talk about here in order to understand this more fully? Let's breed 2 kangaroos. So let's have a male who's heterozygous or normal for the albinism gene. So heterozygous meaning big F, little f. And the female is also heterozygous or normal for the albinism gene, also big F, little f. So we want to know what is the ratio of albino offspring in this cross. So if we made it a big F, little f with another big F, little f, what are the chances of getting an albino kangaroo offspring? And so here I'm just going to set up my Punnett square and we're going to see what happens. So I have the male on top here. We've got big F, little f, and the female on the side here, big F, little f. It really doesn't matter what side you put the different contributors to. I just put the male on the top and the female on the side. So if we were to combine all of these, we're going to see that big F is going to be here, big F, little f is going to be here, big F, little f is going to be right here as well, and then a little f, little f. And so here's our results. And so you just combine these alleles and see what the combinations include. And these Punnett squares can get more complicated as you add certain things, but it's important to understand that all you really have to do is combine one allele with the other in order to understand what the potential outcomes are of the offspring. So when we look at this Punnett square now, we notice that we've got 3 different dominant combinations out of the 4. And so we've got a homozygous dominant, 2 heterozygous dominants, and then we're left with 1 of these homozygous recessives. So we've got 3 out of the 4 of the offspring are dominant and 1/4 of offspring are recessive. So what are the chances? Let's do the math and figure it out. So when we do this math, 3/4 is equal to 75%. And so if you think about it in 100%, if you think about out of 100, you're going to have 75% chance of being phenotypically normal. And then 25% are going to be phenotypically albino. So just having the ratios and then understanding how the ratios relate to the percentages, that's going to help you understand the chances of those 2 contributors having an offspring that is albino. So 25% chance. Awesome. So I hope that that helps a little bit in you guys understanding Punnett squares, how they work, and how you're able to do the math about them. Of course, if you were to go back and add big F as the father and big F, little f, our results would be really different than what we have here. And so you guys are going to be playing around with those different combinations of heterozygous and homozygous individuals to see what the chances of certain characteristics are for offspring. So hopefully that helps all together. But now what I'm going to do, I'm going to stop presenting and I'm going to go into the lab that you guys are going to be doing this week, which is the single cross Punnett square simulation. And so the main question that we're going to be answering in this lab is how well does a Punnett square predict actual outcome ratios? And so you guys are going to make a prediction based on a certain trait and 2 parents that are contributing to offspring and what they code for that trait. And so understanding what their alleles are, making a prediction, and then what we're going to do is we're actually going to come up with the outcome, like what the chances are of actually getting that particular organism with that trait. And so what you guys are looking at is toes. And so you guys are going to be looking at a trait that codes for the short big toe on humans. So when you have a foot that has a shorter big toe and a longer second toe, or a longer big toe and a shorter second toe, just the differences in your feet and how they look. But what you're going to be doing is you're going to be deciding, you're going to have 2 pennies. So I've got 2 pennies here. They're both heads and both tails on the back here. And so you need to decide what side is going to be dominant and what side is going to be recessive. So in my case, what I did is I had heads as the dominant trait and tails as the recessive trait. And so it doesn't matter which side you choose, you just need to be consistent throughout the entire experiment. So you will have a chance to choose on page, I think 2, what side is going to be dominant and what side is going to be recessive. And so the reason why you have 2 pennies is because one of the pennies is going to represent your mom and the other is going to represent your dad. And so these 2 pennies are going to be what contributes the alleles to your genetics. And so these are going to be really important for understanding. And so what you're going to be doing is you're going to be flipping pennies essentially 100 times. And so don't freak out because it only took me about 5 minutes to flip 2 pennies 100 times, but that will allow you to understand the actual probabilities out of 100, and it will make the math a little bit easier. So what I did is I just flipped pennies 1, 2, and what I've ended up here is with a head and a tail. And so if you decided that your head side is dominant, then you would have big T. And if you decided that your tail side was recessive, then you'd have a little t. So my combination of head and tails is big T, little t. And so there is a spot on the 3rd page for you to tally that. Let's say that you got a head and a head. That would be a big T in the case of my choice of sides. And so then I would tally 1 for that side. And then if I got 2 tails, then that would be little t, which I could tally as well. And so you're going to tally up to 100 times, and you'll see that the ratios are going to be different. But make sure that you actually flip it 100 times because it will help with math. So then you're going to total up your tallies, divide over 100, which you'll figure out, and then you're going to compare to the values that you predicted. So based on your Punnett square. And so we'll understand whether or not a Punnett square is a reliable predictor of probability in genetics. And so it's a really cool lab. I think you guys will enjoy it. If you have any questions, please feel free to email me. You guys know my email on the Google Classroom, so please refer to that. At the end of the lab, there's going to be questions that are regarding the lab that you just completed, and hopefully you'll understand how helpful Punnett squares actually can be for scientists to predict potential outcomes of offspring. So hopefully that was helpful for you guys. Like I said, please email me if you have any questions, but hopefully you guys are enjoying the sunshine and getting outside. You can do this lab outside, so have at it. All right, see you guys soon. Hopefully you guys are doing good, and I will talk to you later. Bye.